Welcome. I'm the director of IAS here, and I'd like to tell you that we have been trying these popular lectures to introduce the research work done by the Nobel laureates each year. So I'm not introducing today's speakers. Carl will do that. But I want to tell you that later this week, Professor Bik-Kwoon Yeung will talk about the Physics Nobel Prize for two people who shared it for neutrino physics. I'm particularly happy to announce that just half an hour ago, it was announced that Professor Yeung and the two Nobel laureates this year, and Professor Yifan Wang from China and a couple other people will win the Breakthrough Prize in Fundamental Physics this year. So they will be the first Chinese, first Hong Kong, I may think even the first Asians to win the Fundamental Prize. And of course, there's you have people, many of you are life scientists. Life scientists also, quite a few won the prize. And I think you can look up, each of them get $3 million US. So it's a big prize. So Thursday, besides Luke's talk, Professor Bik-Kwoon's talk, we will probably have a real reception to celebrate his winning the Breakthrough Prize. So I hope you can come, or tell your friends to come. So, okay, Carl.
So it is my pleasure to introduce the person who is going to tell us about this year's Nobel Prize in Chemistry, and that person is none other than our own Bik Tye. Now, you might wonder why Henry Tye was not introducing Bik Tye, and I will let you discuss that with the two of them afterwards. My only credentials in this area is that I do know life science, but I don't know chemistry. And I do know Bik because I am her head. I'm the head of life science. Bik has had a long and illustrious career, beginning with training at both the undergraduate and graduate levels in the United States, at Wellesley College, at MIT, at UCSF, and then for many, many years was on the faculty at Cornell University in Ithaca, New York, where she actually remains an emeritus professor to this day. But in just recent years, she has been gracious enough to join our faculty, first as a professor and now as a visiting professor of life science, and we're delighted to have her. Bik is particularly appropriate to talk about this year's prize because really since her earliest days, she has had her head inside the nucleus of the cell. And inside the nucleus of the cell is where all the DNA lives. And so it has the chemistry of DNA has been an issue that has really intrigued her and powered a hugely successful career in molecular biology for many, many years. Her interests have been largely in how DNA replicates itself and makes more of itself. The prize this year is more about what happens when the copies get broken and need to be fixed. But it is certainly the case that we have an expert in the field and one who is eminently qualified to tell us about this year's Nobel Prize in Chemistry. So I give you Bik Tye.
Well, first of all, I want to congratulate Bik-Kwoon for his prize. And I look forward to that reception that Henry promised. I have everything here. Okay, this year's Nobel Prize in Chemistry was awarded to three people: Thomas Lindahl, Paul Modrich, and Aziz Sanja. I think this is how it's pronounced. So this prize was awarded for their work on mechanistic studies of DNA repair. Thomas Lindahl is a Swedish-born British scientist. He's now a Professor Emeritus at the Cancer Research, UK. Paul Modrich is Professor at Duke University, HHMI Professor. And Aziz Sanya, he is a Turkish-American scientist, and he's a professor at the University of Carolina. And the three of them share the prize equally. So each of them did a very different piece of work, but they're related. So it all has to do with how do cells take care, maintain, and repair damages that incur on our DNA. For Thomas Lindahl, he showed that DNA molecule, our genetic material, is actually not that stable. It undergoes spontaneous decay at the tune of 200 damages per cell per day. And his work basically showed that there is a special mechanism called the base excision repair mechanism that takes care of this particular kind of damage. For Paul Modrich, he's interested in understanding how during DNA replication, we have huge genomes, and to replicate every single base in our genome correctly is a very meticulous task. And he showed that mistakes made during DNA replication, which occurs about 1 in a million, is taken care of this repair mechanism called mismatch repair, or MMR. For Aziz Sanya, he showed that our DNA is subjected to external insults such as UV, irradiation, cigarette smoke, and whatnot. And that also incurs damage to our DNA. And he showed that this kind of damage is repaired by the pathway called Nucleotide Excision Repair, or NER. So 3 different tools repair 3 different types of DNA damage. And this is actually a very nice crowd. All 3 of them are very low-key, collaborative co-laureates who talk to each other, friends with each other, and they work on different mechanisms that now actually kind of merging. Okay. All right, so let's, first thing first, let's give you the background. So our genome is made of chromosomes, and chromosomes made of DNA that are packaged into these tight structures, compact structures called chromosomes. And the DNA helix is a double-stranded DNA, and it contains 4 bases, A, T, G, C, thymine, adenine, guanine, and cytosine. Thymine must pair with adenine, guanine must pair with cytosine. So we have 46 chromosomes, and we have 6 billion base pairs in our genome. We're talking about a lot of DNA. So when the cell divides, the DNA has to be replicated, and the duplex DNA has to be unwound. The replication machinery that would unwind the duplex DNA so that each of the single strand, the parent strand, can serve as the template for the replication of the new DNA strand. But during this process, it is really imperative that A again pairs with T, G pairs with C, so that the information that was in the original copy is passed on to subsequent divisions. Okay, so actually I should mention here that my group here, which is now sitting right here, Yeung Tye and Philip Yeung, we are working on the replication machinery, and in particular, the part that unwinds DNA. Okay, so what ties these 3 scientists together? They all, their research is driven by curiosity. This is truly curiosity-driven research. They have a very simple questions, and they set out to ask the address the question by using the simplest model system that they can find, and that is E. coli. So their initial works were done in E. coli, and they found the enzymes that repair DNA damage in E. coli. Later on, they found that they're also involved in the repair in DNA repair in human. And then now they actually show that human cancers are due to mutations in some of these DNA repair genes. And not only that, the DNA repair pathways, while important for maintaining the integrity of the genome, are now used as targets for chemotherapy. Because cancer cells are basically due to the accumulation of mutations that cause the cell to proliferate uncontrollably, and each one of them undergo uncontrolled cell proliferation for different reasons. All depends on the combination of mutations. So to a certain extent, the defect in DNA repair pathway would result in the accumulation of mutations. But if you have too much mutations, if there's too much damage, the cell actually would die. So one of the targets for chemotherapy for cancer is actually to make the repair system so inoperable that the damage actually kills the cancer cells. So this is how this field is developing. Okay, so I just want to give you some preview of the next, or maybe the next Nobel Prize to be awarded, because just this year, the 2015 Lasker Award was awarded to Evelyn Witkin of Rutgers University and Steve Elledge of Harvard for their work on DNA damage response. So this is, again, a whole new field, basically addresses how cells go through a series of measures to temporarily halt DNA replication and cell cycle progression when it detects that there is damage in the DNA to ensure proper repair. So this is another whole DNA repair field that is really gaining track. Okay, so I'll first talk about Thomas Lindahl. He was born in 1938 in Stockholm. He's a naturalized British citizen. He did his PhD and MD from Karolinska Institute. And even for his PhD thesis, he was already interested in the stability of nucleic acids. His thesis was on the structure of nucleic acids in solution. As a postdoc in Princeton, he was wondering how stable nucleic acids it was, and he was looking at RNA. It turned out RNA was extremely unstable, and then he turned to look at how stable DNA was, and this led to his current work. He was a professor of medical chemistry in the University of Gothenburg from 78 to 82, then went to Imperial Cancer Research and was director of the Clare Hall Laboratories, and he's now emeritus group leader of the Francis Crick Institute. And I took this picture of him from the news, and what is shown here is him and his granddaughter, and it made news because his granddaughter reported to her teacher that she couldn't hand in her homework because her grandfather won the Nobel Prize the previous evening, and she had to celebrate. So this is very interesting. Okay, about curiosity-driven research, Thomas Lindahl basically asked a very, very simple question. Our DNA is a chemical molecule. How stable is it? And he basically showed that DNA decays at a rate. Oops, this is really. Decays at a rate that ought to have made the development of life on Earth impossible. This insight led him to discover a molecular machinery called base excision repair, which constantly counteracts the collapse of DNA. Okay, so what kind of spontaneous decay are we talking about? The most common ones, so we have spontaneous oxidative damage, which is shown by the red arrows, hydrolytic damage, shown by the blue arrows here, and then uncontrolled methylation or alkylation. But the most common ones that are are really shown here in the blue. The predominant hydrolytic damage, the deamination of cytosine, the removal of this amino group would turn this cytosine base into uracil. And then the hydrolysis of the glycosidic bond that links the base to the sugar moiety. So these are the kind of decay that our DNA is subjected to daily. In fact, now, while you're sitting here. Okay, what is the consequence if we don't take care of these spontaneous mutations? So as I mentioned, the common hydrolytic of the deamination of cytosine. So if cytosine is deaminated, the amino group removed, is turned into the base uracil. But uracil does not pair with guanine. So if you allow this mutation, this damage to persist, it will be replicated. So U would now pair with A, so, and basically it sort of fixes a mutation, okay? So this damage has to be repaired before it's being fixed. Now, the other type of hydrolytic damage is depurination, basically the removal of the purine base from the phosphate backbone. And what happens when you have a depurinated purine? If you remove an A, there's an empty space here, so when this strand is replicated, this A would be deleted, so you have a single base deletion. And of course, this strand is okay. This strand, nothing will happen to it. But again, if you don't fix this problem right away and allow the DNA to be replicated, you get a mutation, a single base deletion mutation. Okay, so this is just a model of showing what happens when you have a deaminated cytosine that's turned the uracil, turn it into uracil, and uracil does not pair with A, so it is unpaired base. So it's actually very easily recognized by this enzyme called uracil N-glycosylase, which will remove it from the unpaired bases. Okay. So it turns out that there are a number of modifications of these bases due to spontaneous decay, and there's a whole battery of glycosylases that would specifically remove that damaged base. So this one is called uracil N-glycosylase. So how does the mechanism work? It's very simple, really. What you see here is it's double-stranded DNA. So you can see here the sugar phosphate backbone, the paired bases, and suppose you have a deamination of the cytosine, you end up with a uracil, and the uracil is no longer paired. Then here comes this enzyme called uracil glycosylase that will remove this uracil from the unpaired base. But there's still a hole here, so something else has to happen. And what happens is that this apurinic site, there's no base here, is recognized by an enzyme called AP endonuclease, apurinic endonuclease, and it will cleave the phosphodiester backbone, and then DNA polymerase 1 will come in and seal the, fill in the gap, but basically it goes through a Nick translation, for those who know what it is. Basically, it will repair because it's a DNA polymerase, it can insert the base pair, the base that will pair with the template strand, and then DNA ligase will seal the nick, and then now you have the damage repaired. So this pathway is known as the base excision repair pathway. Okay, so are there questions about base excision repair? So that's one pathway. This pathway only takes care of the spontaneous decay of the bases. And the last estimate I read somewhere is about 200 bases per cell division. We're talking a lot of damages. Okay, so now let's go to Paul Modrich. Paul Modrich, actually, he's the first he's the son of immigrant parents from Croatia and Montenegro. So he's the first generation scientist here in the US. Was born in New Mexico, got his bachelor's degree at MIT, then went to Stanford for his PhD. And he worked with Professor Bob Lehman, who also was my postdoctoral advisor. He graduated in 73 and I arrived in 74, so we did not overlap. But nonetheless, we met from our laboratory gatherings and when he visited Stanford. Well, that was a time when recombinant DNA was brewing, and he was in the right lab. In fact, the whole department basically works on DNA, pretty much. And he worked with DNA polymerase and DNA ligase and also ECOR1. So he was very familiar with enzymes that replicate DNA. And this has something, it sort of lays down the background for his subsequent work. Then when he became a postdoc, he went to Harvard, and there he met Matt Methyl, who, if those of you who took biochemistry course, you know that Methyl installed the same Methyl. He was doing some experiment where he made a heteroduplex of a viral DNA, one containing the wild type strand, the other with some mutations. And when he transfected that into E. coli, somehow the mutations were repaired. And he couldn't figure out why was that repaired. And anyway, he had some idea that it may have to do with when he was making the heteroduplex, one of the strands was methylated and the other was not. And the one that was methylated directs the repair of the mismatched bases. So when Paul Modrich arrived, he was working with an enzyme in E. coli called the DAM methylase, and I will talk about it in a minute. And that is basically he was there at the right time, at the right place, with the right hypothesis, and he was going to test it. And that sort of developed into this wonderful study that he ended up doing. He became assistant to full professor at Duke, and HHMI professor, and so on. And what is shown here is, of course, for those of you who know, who have been in the US college basketball scene, Duke is a strong power is a contender in the basketball. What league is that? I don't know. But anyway, he was honored with this his own jersey by the head coach. So clearly, everybody was very happy, including the basketball team at Duke. Okay, all right, so let's talk about Paul Modrich's problem. And that is when our genome is replicated, how many mistakes does the DNA polymerase really make? So let's look at the size of our genome. It's 6 billion in the human genome. And the estimate, it is very rough, is we have 4 times 10 to the 13 cells in the human body, and they all derived from a single cell. We all came from a single cell. So that means that we came from the result of 2 times 10 to the 13 cell divisions. That's a lot of replication, right? So any mistakes made during DNA replication is a mutation. The DNA replication has to be very accurate. The overall error rate for DNA polymerases with inherent proofreading function is about 10 to the minus 6, 1 in a million in the test tube. But the estimation is that, well, maybe in the cell is actually better, but still couldn't be more than 1 in 10 to the 8th. So this is somebody coming from a background where Stanford biochemistry, where DNA polymerase is studied. And we all know that for the DNA polymerases that replicate the genome, not only that the pairing of the base pairs is very, very important, but also once the polymerase makes a mistake, there's an inherent proofreading function that corrects the mistake. So even so, it still makes about 1 in 10 to the minus 6 to 1 in 10, 1 in a million to 1 in 10 to the 8th. So the hundreds or even thousands of mistakes made every cell division due to just DNA replication, just replicating the DNA. So the question is, suppose an error has been introduced during DNA replication, and suppose there is a repair mechanism that can correct that mistake. Paul Modrich asked the simple question: how is the parental strand distinguished from the daughter strand that contains the mistake during repair of mismatched bases? If the repair mechanism cannot tell which was the original base and which was the mistake, it could actually fix the mutation rather than correct the mutation. So that was the question that he asked, very simply. And remember, he was at Harvard, and Matt Methelson was talking about, well, there's something funny about transfecting virus DNA into E. coli. Somehow the heteroduplex mistakes are repaired. So then he tested the model, and since he worked with DAM methylase, he could make any kind of heteroduplex at will in vitro. But anyway, this is how E. coli does it. So the DAM methylase is an enzyme that recognizes the sequence GATC, which is a palindrome, which reads GATC on the other strand, and it methylates the A. So during DNA replication, the new strand is being polymerized and copied from the original template. But for a short period following replication, the template strand is methylated and the new strand is not. And this is only a very short time. So at a during a brief few minutes, probably, the newly replicated DNA is hemimethylated. Only one strand is methylated, the other is not. So, and then after that, the DAM methylase comes in and methylate the other strand. So there's a very narrow window of opportunity when DNA repair must take place in order to correct the mistake that is incorporated into the new strand. So this is what was found, that during DNA, when there is a faulty base, suppose this is a mismatched base pair here, and that the methylated strand is the parental strand and the copy strand is not methylated. And during this very narrow window of opportunity, two protein factors, MUT-S and MUT-L, bind to this mismatched base pair. And then another factor called MUT-H binds to the nearest, binds the methylated site. And by bending, these factors actually can interact, and upon interaction, MUT-H would act as an endonuclease and cleave at around where the methylated base is here, and then now this copy is cut, and then again DNA polymerase would come in, fill in the gap, and actually DNA polymerase. Remember, DNA polymerase 1, while it is filling in, it can also remove the damaged base, and the error is corrected, and ligase seals the gap. Okay. So I can actually show, we have lots of time, so might as well show you an animation so that you can see how it works. Okay, so let's see. This is how.
Incorporation of base pair mismatches during DNA replication does occur. There are DNA repair mechanisms that can fix such mistakes. One such mechanism is the methyl-directed mismatch repair system. In Escherichia coli, this repair system can detect a mismatch shortly after DNA replication and repair the newly synthesized strand, thus preserving the base pairing that was present in the original DNA molecule. The system relies on the fact that methylation of a newly synthesized DNA strand does not occur until several minutes after the strand is made. This provides a window of time during which the newly synthesized daughter strand in a DNA double helix is distinguished from the parental strand, which is already methylated. A sequence of DNA with only one methylated strand is said to be hemimethylated. Actual recognition of a base pair mismatch is the role of the Mut-S protein. Mut-S binds to a mismatch site and forms a complex with another protein, Mut-L. The formation of this complex recruits Mut-H to the region. Mut-H binds to a hemimethylated site. The Mut-S, Mut-L complex interacts with Mut-H by a DNA looping mechanism. Mut-H then makes a cut specifically in the non-methylated strand. An exonuclease digests the non-methylated strand from the cleavage site to just beyond the mismatch site. This leaves a gap in the daughter strand, which is filled by DNA polymerase III and then sealed by DNA ligase. The end result is that the mismatch has been replaced with the correct base pair as indicated by the original DNA strand.
So that's the mismatch repair mechanism. Let's see. Okay, so that was in E. coli. And of course, the E. coli is the model system, and once you know how E. coli works, you basically reproduce the experiments, maybe in the more complicated system, and human was actually a very good system to look at for in vitro studies. And basically, Paul Modrich, of course, it's not just his lab at this point. There are really a lot of people also looking at this system. Basically, a human also used the same set of enzymes. However, methylation does not play a role in directing strand-specific repair. In fact, we don't know how human or in high eukaryotes, how do they figure out which is the right strand to repair from. Okay, so the main points, the mismatch repair, Paul Modrich brought it from E. coli to human. He's now able to reconstitute the human mismatch repair system with purified proteins in vitro. So we're really at the state that we can understand mechanistically how mismatch repair is carried out in E. coli as well as in human. It's still unknown how the strand-specific repair is directed in the human. That is still a big question. What is comforting is that there are conserved homologs of Mut-S and Mut-L, and of course in human there are many additional factors, but Mut-S and Mut-L are conserved. And the importance of MMR in human is underscored by defects in the pathway that cause hereditary non-polyposis colon cancer. So basically, this tells you that the study that he started out by asking very simple questions that he did in E. coli and now showed homologous proteins in human, it actually played a very important role in one type of cancers. It's a hereditary cancer called non-polyposis colon cancer. People who inherited these mutations in genes in this pathway are prone to develop this type of colon cancer. So clearly, this fundamental mechanism tells us a lot about not only about how the cell works, but how we can use it to study cancer and devise therapeutics to cure cancer. Okay, so now, are there questions about the mismatch repair? Yes.
Mut-L and Mut-S complex together, and it says that complex recruit Mut-H to bind to the methylated A. So I wonder how the mutant L and mutant S recruit the mutant H from another site, and then pass the loop.
Oh, so how do they find each other? Okay, so that is a question that people often think about, and this is not uncommon that DNA that are bound, in fact, in enhancers, in promoters, and so on, that factors that are far away will come together. So presumably, DNA is not static. They are actually moving around, except that it is not in free diffusion, that when they bound to two sites, they can still, in fact, it sort of limits the distance that it may increase their chance of finding each other. Is that what you're asking?
Actually, no. So my question can be explained why the mutant H bind to the methylated A, right? Yeah. So is it depend on the mutant L and the mutant S bind to the mutation site first?
Yes, I did.
Always bind to the...
Yeah. I know that it is, you have to remember that it's just a cartoon, okay? It's just to help people to see, but the mechanism is much more complicated. It's not clear that these are independent events. They are, there may be signals of one bound to the actually it's quite far away where they're bound, far enough that they, so I'm not sure that, I'm not in the field, I don't know if they know what is the mechanism that allow this complex to be bound at two different places and come together. But there are quite a few of these kind of interactions that I'm not sure that we know the answer to what kind of signal it is sending from something bound far away to come together. But that's a question I think a lot of people are thinking about, unanswered for a lot of these complexes. Okay, are there other questions?
You just mentioned for human that you cannot distinguish between the old and the new strand. But
well, they can obviously, but we don't know how it does it.
also, you don't find this protein which bind to the.
So there are some speculations. One is that it has to do with PCNA, and that it uses a lagging and leading strand as a way to distinguish. But I don't really see how that works out. It's just a hypothesis. I don't think it's at the stage that people can say, yes, this is how it works.
Can they use the same idea as that the methyl group on the A and...
Well, I'm sure there is something that people, clearly since it works that way in E. coli, then people assume, well, maybe that's... but it turns out that for high eukaryotes and for human, DNA is methylated all over. And there are many different kinds of methylation. And there's one, it's quite possible, there's one type of methylation that we are not aware of. It's what's distinguishing it, but it's not found. People have not been able to identify. Human DNA is methylated, modified by many, many, many different kinds of adducts. And we still don't know all their functions. And I think people like Danny one of these days will tell us how some of them work. But people have looked very carefully at methylation. Okay, so now we go to the work of Ashish Sanya. So actually, I was looking at all the images about Ashish, and clearly Turkey is really, really, really happy to have their first Nobel laureate. Actually, Ashish Sanya was also the first person to get into the National Academy, first Turkish American to get into the National Academy, first of many things. But his struggle, maybe he didn't think of it as a struggle, but if you look at how he got to where he is, it was not simple. It was not easy. So Ashish Sanya was born in Turkey. He was the seventh of 8 children of illiterate parents. But even though his parents were illiterate, they valued education. So all their children had good education. That was a very important point. He was actually very good at soccer, especially as a goalie, and he was drafted to play professional soccer before he went to medical school, but he decided that he was not tall enough to make it. So he decided to go to medical school. He went to Istanbul University, and then he decided that he had to go to a place where the actions are, and he decided he should go to the U.S. And I think I read somewhere that when he was trying to move to the U.S., at that time, John Lennon also wanted to go to the U.S. but was barred because he smoked marijuana or something. And they asked John Lennon, why do you want to go to immigrate to the United States? And he said, well, if you live in the Roman Empire, you would go to Rome. And this is the time when the U.S. was extremely powerful and popular. So everybody wanted to go to the U.S. And he made that comment and he said, well, he decided he had to go to the U.S. because that's where all the actions are in science. So he went to UT Dallas. And he was interested in photoreactivation, in particular this enzyme called photolyase. Basically, in E. coli, if you expose E. coli to UV light for too long, it will kill the cell. But if you expose to UV and then let it sit in longer wavelength light, such as blue light, it will reactivate and the cell is fine. But if you leave it in the dark, it will, if the dose is too high, then so there's this enzyme called photolyase, and he was very interested in it. But in the end, his whole project is looking at UV damage. Turns out photolyase is only present in E. coli or in other systems, but not in human. Okay. But E. coli, besides photolyase, there is another dark UV repair pathway. There's a slow pathway. And that's the pathway that he basically focused on, and this is what I'm going to talk about. Okay, but this is how he started. He was looking at the photo reactivation. Okay, so by the time he graduated, he wanted to look for postdoc. He applied to 3 places, and he was rejected in every place. But his wife-to-be was going to Yale, so he found somebody who has a vacant technician job. So he went to, so this person, Dean Rupp, wanted him, but he only had the technician job. So he went, even with a PhD, as a technician. And he worked there for quite a few years, but it was a good choice because Yale at that time, there were people like Howard Flanders and Charles Redding, who were really into looking at DNA excision repair. And there he cloned UVRA, UVRB, UVRC genes, which are the enzymes that in the end he called them excinuclease, that is responsible for repairing UV damage. He went from being a sort of postdoc to become in one step as associate professor at North Carolina because his work clearly showed a great promise. And then later he showed that humans use the same mechanism to excise thymine dimers. But what is interesting is that the human enzymes are not conserved. They have no homology to the E. coli enzyme. So this mechanism basically is evolved by convergent mechanisms, which is quite amazing. But anyway, this is how biology works. When the cell needs something, they'll figure out the way to do it. Even though it's the same strategy, they'll evolve to do it. All right, so what exactly is this UV damage? When we sit in the sun, our DNA is exposed to UV light, and UV light can cause these kind of covalent linkage to thymine dimers. So if there are 2 adjacent thymines here, UV light can result in the formation of cross-links between these two bases, thymine bases, to form cyclobutane thymine dimers, or this kind of photoadducts. So when that is formed, they can no longer pair with the A's, the opposite bases, and they form a kink. So it forms a very special structure that clearly the repair enzymes would recognize. So, all right. So in E. coli, when you take irradiated E. coli cells and exposed to visible light, there's a spontaneous photoreactivation. There's the enzymes called DNA photolysases that can restore this, break up this bond. But this is not present. This system is not present in human or in mammalian cells. Okay. So there has to be another mechanism, and this is what he basically looked for, dark repair not in light. And this is what he found, the nucleotide excision repair mechanism. And here I will give you the animation, which I can't enlarge it. I hope it's visible to you.
Nucleotide excision repair is an important general process that cells use to correct many different types of DNA damage, including UV-induced thymine dimers. Although present in all eukaryotes and prokaryotes, the molecular mechanism of this system is best understood in prokaryotes. This animation will illustrate the process of thymine dimer repair in E. coli. The nucleotide excision repair process uses a set of proteins called UVRA, UVRB, UVRC, and UVRD. UVR stands for ultraviolet light repair of pyrimidine dimers. But remember that this system is also used to repair other types of DNA damage. In addition, DNA polymerase and DNA ligase are used to complete the repair. The process begins with the loading of a protein trimer onto the DNA double helix. This trimer consists of 2 UVRA molecules and 1 UVRB molecule. This trimer moves along the DNA, scanning for damage that distorts the double helix. When the trimer encounters DNA damage such as a thymine dimer, it stops. The UVRB protein remains at the damaged site, while the 2 UVRA proteins are released. UVRC then binds to the site and makes 2 cuts on the damaged strand. Both cuts are several bases away from the damaged site. UVRD, a helicase, then binds to the site and separates the segment of damaged strand from the rest of the DNA molecule. UVRB, C, and D are then all released from the damaged region. This leaves a gap that is filled with the proper nucleotides by DNA polymerase and then sealed by DNA ligase. The end result is that the damaged DNA has been replaced with the correct bases.
So that was the animation. Again, this is a cartoon, so it's much more complicated than what it's made to be. The purpose is to understand the importance of the work and what role these repair pathways, these tools do. Now, what you just saw in the animation was that of E. coli. And I told you that human also uses the same mechanism, but by convergent mechanism. And what is amazing is how similar they are, all right, by convergent mechanism. Here in E. coli, as the animation told you, that the enzyme UVR-ABC, called hexanuclease, binds to the kink, and it cleaves a 13 base sequence, and then DNA polymerase 1 ligase fills in. For human, there's also an hexanuclease, but it's totally non-homologous to the E. coli enzyme. It also cleaves a piece out, but it cleaves out 29 bases. and then uses DNA polymerase epsilon and the DNA ligase to fill the gap. So this is another remarkable thing about what he found. And what I want to tell you about is how these very fundamental studies, starting from E. coli, led to human and actually allow us to understand about diseases, is that a human genetic disease called Xeroderma Pigmentosum, it turns out it is a defective in enzyme for pyrimidine dimer excision. And these are patients with this kind of genetic mutations will lead to multiple skin cancers. So here's a sort of a picture of somebody who has a mutation in the XP pathway. They are extremely sensitive to sunlight, develop skin cancer, even upon short exposures to sunlight. But what's also interesting is that they also have neurological abnormalities. People don't think that this neurological abnormality is due to exposure to sunlight, but rather that this pathway, this NER pathway, probably also repair DNA damage of other kinds. And that's why, even in the part of the body that is not exposed to UV, you still have these severe damage. Okay, so I think that concludes what I have to say. And I will go back to just summarizing for you that we have 3 Nobel laureates, newly minted Nobel laureates, asking very simple, fundamental questions, use E. coli as a model system, and just follow their curiosity and chase down what exactly is happening. And they found out these very important DNA repair pathways. Each of them take care of one particular type of damage, but actually it could be more. We don't know what other damage it will repair, but clearly a significant number. But that's not all. I want to tell you that this is just 3 of the tools within our toolbox for our cell to repair our DNA. There are other ones that we may not have found yet, and we know that they're there because there's something there. We know that there are other repair mechanisms going on. So 3 different tools to repair 3 different types of DNA damage. That's what the 2015 Chemistry Prize was awarded for. Okay, so questions? I don't necessarily have the answer for you, but I'd be happy to.
I have a question regarding Xeroderma Pigmentosum, I think the name was called, because you mentioned how that might be, the protein might be related to other types of DNA damage response, which might be the reason why, if there's a mutation in that protein, that you get some sort of neurological disorder. So is that sort of neurological disorder because of cell death due to chromosomal instability, or have they figured out what they're doing yet?
Actually, I think that's something that Carl works on. He might want to answer the question. But what I was saying, really, but I'll let Carl answer. But what I'm really saying that this pathway, which is called the nucleotide excision repair, we only studied it in terms of UV damage, how it repairs UV damage. It's possible that it repairs other kinds of damage. And why there are more damage, or why DNA damage is more sensitive or exhibit, give us these neurological diseases, Carl can answer, I think.
No, I can't.
You can't?
Well, what is known is that, 2 things. First of all, because nerve cells never divide, they are missing a whole repertoire of DNA repair mechanisms, not these. So they're much more sensitive to DNA damage. But the other thing which is relevant, as students in 1902 will all remember, is that almost all the DNA repair defects are characterized by premature aging. so that if you can't repair your DNA, your cells eventually lose the ability to function normally, and so they die. But we have a question in the front here.
What happens if there's two DNA errors?
Okay, Preston's question is, what happens if there are two DNA errors from the same kind of error or different types of errors? They are all working at the same time. These tools, they're not like you go first, I go second, they're all working at the same time. So they will all...
Then what happens if they're both the same one and they're both very near each other?
Oh, you mean would they be fighting to repair for one particular damage? Is it mine or yours damage? Who takes care of it? Is that what you mean?
I mean, like, if there's like two different damage or two the same damage in one same like area, like very close to each other. Then how do you do it together?
Okay, he's asking if there are two similar or same damage close to each other, how do they repair it? Well, actually, that is interesting. But okay, when does that happen? Okay, let's say there's a UV. You're saying there's a pyrimidine dimer that is close to a DNA mismatch error due to DNA replication. Is that right? Okay, so the first thing probably the mismatch repair. It turns out that as the person up there was saying, that the enzymes that recognize the mismatch and the methylated strand, they could be very far away. So actually that is a very expensive repair mechanism. So what happens is that actually it takes out the whole piece and fill in, could be up to a kb or more of DNA, thousands of bases. So it may take care of the close by whatever other damage there is. So we don't know why that particular mechanism would be so expensive, so costly. It has to, if there's a mismatch due to error incorporated by DNA polymerase, why should the whole piece be taken up? Okay, so that might be one of the take care of your concern. The other thing is a probability thing. It's the likelihood of two pyrimidine dimers right next to each other. Or, you do a calculation of how often does this kind of cross-linking take place. The likelihood, but I don't think it will. So they take care of one and then the other. That shouldn't be any kind of issue. But that's a very good question. Thank you.
Okay, this prize is given for discovering 3 different tools in the toolbox.
Right.
But I suppose the DNA damage, there are more than 3 different types of DNA. That will be quite a lot.
That's
so
right.
At this point, for the molecular biologists, how many different type of different kind of DNA damage, and how many tools do we have in this toolbox? Roughly.
I don't know. We make it sound very specific. There are only 3 types of damage, then 3 types of tools. But actually, we define the damage based on the initial study, for example, as I mentioned, the nucleotide excision repair. There are neurological problems that has nothing to do with UV damage. So clearly, the same tool, actually, we define it as repairing just thymine dimers. It probably repairs a lot of other things. But how many there are, there must be other ones. I don't know how people figure out, but there's the whole, as I mentioned in the beginning, there's the whole idea about DNA damage response. The cell has to do, I don't know if we know all the things the cell have to be, basically in the Lasker Award that was awarded to Steve Ellich, and Evelyn Whitcomb. In that case, when the cell senses there is a broken DNA strand, there's no point to go on any further because one break in the genome it will cause death. All right, so it doesn't. What the cell would respond to is arrest everything in the cell, no replication, no more cell cycle progression, and make sure that the repair system goes there and repair. And that we know is recombination. They use a totally different kind of repair, recombination repair, and that even the recombination repair can be branched into many different things. So in that system, we use the cell uses another set of tools with another kind of toolboxes. Well, we have a large toolbox, but what is amazing is that how DNA turns out it's not as stable as we conventionally thought of. It turns out it's really subject to a lot of insults from the environment, from spontaneous decay, from its own replication. But we have evolved to be so good at taking care of problems to make us, it is really a miracle that we can walk around and last for so long. Because these DNA repair tools have to be working full time to take care of all the damage. However, we have to have some errors. Otherwise, there will be no evolution because we will all remain the same forever. Because there's a balance between mutations and repair that allows evolution. And just the right amount in the right sequence in the right environment that allows the evolution of complex organisms.
So you mentioned before that sometimes the repair can be very expensive because of you need to cut a very long chain. So my question is quite similar to that boy's. So now I wonder if in MMR mechanism, there is one error, but two methylated. So one may form a very large ring. I need to cut a lot of DNA. Another one, I may form a relatively smaller ring. So is there any observation that it's just randomly forms the ring, or preferably forms a small ring because I just need to cut a very small amount of DNA and
okay, yeah,
ATP.
So the methylation. This is in E. coli. The GATC is a 4-base recognition for the DAM methylase. So its occurrence frequency is probably 1 in 256 to 300 bases. That's how frequently that site is. So if they are all methylated, and that when you have a mismatch, then the closest ones are they're not. It's not like you have to find a methyl group far away to repair this, because these signposts are all over scattered on the genome, spaced about 300 base pairs apart. So does that answer? So it's not that costly. Maybe we're talking about a kb at around on the average. That's how long it has to.
My question is that will favorably choose one methylated compound? For example, I have one.
So
right to my.
So that also addresses the other question. I actually don't know, but it all depends on how many of these MUT-H proteins. If they're very abundant, they can occupy every single methylated site and be available for the closest S-MUT-L complex. So you have to think about the concentration of that particular protein. If MUT-H is high enough concentration, maybe they're binding to every methylated site.
So it's binding by chance?
I don't know. I actually don't know that. I didn't see it in the literature about it. But if you think about it, but people do know that length of repair is about 1,000 cases.
Right.
Okay.
If there are no further questions, please join me in thanking Professor Tye for a wonderful summary of this year's prize in chemistry.